Hello guys, welcome back to our channel. Today we will be discussing about the de novo pathway for the synthesis of purine nucleotides. So here we will be first discussing about the formation of IMP or inosine monophosphate, which is the first part of the pathway, and in the next video we will be discussing about the rest of the pathway for the synthesis of purine nucleotide. So here the formation of IMP or inosine monophosphate starts with ribose 5-phosphate. This is the ribose 5-phosphate. This is in alpha form, that is the OH group attached to the first carbon is in the same side as that of the other CH2 OH groups. Thus this is the alpha form of ribose 5-phosphate, and this ribose 5-phosphate in presence of phosphoribosyl pyrophosphate synthase or prpp synthase forms phosphoribosyl pyrophosphate or prpp. So what happens here is this ATP molecule gives its 2-phosphate group to this ribose 5-phosphate and gets converted to AMP. So this 2-phosphate of ATP joins with this oxygen and forms this bond here. Now the remaining hydrogen, that is this hydrogen of this OH group, leaves with AMP. Thus PRPP is formed. This is the first step. In the next step, this PRPP in presence of glutamine PRPP amidotransferase forms 5-phosphobeta-D-ribosylamine. So what happens here is this much group leaves as PPI. This much group leaves as PPI. Then this glutamine donates its amido group to this carbon, first carbon, and leaves as glutamate. So structure of glutamine is COOH, CHNH2, CH2 twice, then CONH2. So this is the structure of glutamine, and here this NH2 group is donated to this first carbon and this CO is left here. So this water molecule, which can be written as H and OH, donates its OH group with this CO and forms an OH group there. Thus glutamate is formed. So this glutamine donates its amino group to this first carbon of PRPP and this glutamine leaves as glutamate after the donation of amido group. So here the remaining hydrogen leaves with this phosphate group, this much group. So first of all, what happens here is this much group gets cleaved and leave as PPI. So this hydrogen of water also leaves with this phosphate group. Then what happens is this glutamine donates its amido group to this first carbon and leaves as glutamate by accepting this OH group of water. Then there is an inversion of this first carbon, that is this hydrogen comes down and the amido group goes upwards, that is there is an alpha beta inversion there. So now this alpha form is converted to its beta form. This 5-phosphobeta-D-ribosylamine is formed. 5-phosphobeta-D-ribosylamine thus formed, then in presence of GAR synthase or glycinamide ribosyl 5-phosphate synthase forms glycinamide ribosyl 5-phosphate or GAR. So what happens here is glycine comes and joins with this 5-phosphobeta-D-ribosylamine and forms this compound. So here this OH group of this glycine, this OH group of the COH group of this glycine and one of the hydrogen of this NH2 leaves as water molecule. Then the remaining CO attaches with this NH group and forms this bond here. Thus GAR is formed. Then this GAR in the next step in presence of GAR formyl transferase forms formyl glycinamide ribosyl 5-phosphate or FGAR. Here transfer of formyl group or CHO group from N-tan formyl THF is happening here. So what happens here is this much group, that is this much ribosyl 5-phosphate group, we write it as ribose 5-phosphate without writing its structure for our simplicity. Now the remaining portion is rearranged here, that is NH, C double bond O, CH2, NH2. NH2 is written as NH here, that is this one of the hydrogen of this NH2 leaves with this tetrahydrofolate. Thus NH is present here. Then the formyl group that is donated by this N10 formyl THF attaches with this nitrogen and forms this bond here. Thus FgAr is formed. Now this FgAr in presence of FgAm synthase forms formyl glycinamidine ribosyl 5-phosphate or FgAm. So what happens here is glutamine comes and donates its amido group to this FgAr and leaves as glutamate. So here this O along with one of the hydrogen of the amide group donated by this glutamine leaves with this glutamate, and this NH forms a double bond with this carbon here and forms this double bond NH here. That is, this O along with the amide group hydrogen, one of the hydrogen of this amide group leaves with this glutamate, and the remaining NH joins with this carbon as double bond NH to preserve the valency here. Thus this FgAm or formyl glycinamidine ribosyl 5-phosphate is formed. The FgAm thus formed, then in presence of AAR synthase or aminoimidazole ribosyl 5-phosphate synthase forms aminoimidazole ribosyl 5-phosphate. So what happens here is energy is required for this step. So an ATP is converted to ADP and PI, and there is water removal here. So here water is removed by this hydrogen from this NH and this hydrogen from this NH, and this oxygen from this CHO group leaves as water molecule, and there is a bond formation between this nitrogen and this carbon here. So in order to preserve the valency, there is a double bond formation between this nitrogen and this carbon here. There is rearrangement of hydrogen atom between this carbon and this nitrogen, and thus NH2 forms here and a CH is formed here. Thus here also, to maintain the valency, a double bond will be formed in between this carbon and this carbon. Thus aminoimidazole ribosyl 5-phosphate is formed. Then this AIR in presence of AIR carboxylase forms carboxyaminoimidazole ribosyl 5-phosphate or CAIR. So what happens here is this carbon dioxide comes and joins with this AIR. So here the CO2 comes and joins with this carbon, and C double bond O is formed here. Then the hydrogen, this hydrogen of this carbon attaches with this oxygen of CO2 and forms an OH there. Thus COOH is formed. Thus CAIR is formed from AIR. In the next step, this CAIR in presence of SACAIR or aminoimidazole succinyl carboxamide ribosyl phosphate synthetase forms SACAIR or aminoimidazole succinyl carboxamide ribosyl 5-phosphate. So here this aspartate. So aspartate comes and leaves as water molecule and leaves a water molecule by joining with this CAIR. So what happens here is there is energy needed for this step. So ATP is cleaved to ADP and PI. Next this MECH group, this NH group, this NH2 group of this this aspartate joins with this carboxylic acid group and forms this compound. So what happens here is there is removal of water molecule. Thus one of the hydrogen of this NH2 along with the OH group of this carboxylic acid group leaves as water molecule. Thus a bond is formed in between these two, and this is the bond here. Thus SACAIR or aminoimidazole succinyl carboxamide ribosyl 5-phosphate is formed. The SACAIR thus formed, then in presence of adenylosuccinate lyase forms AICAR or aminoimidazole carboxamide ribosyl 5-phosphate. So what happens here is there is cleavage of carbon nitrogen bond, that is there is cleavage of this bond. So now this hydrogen joins with this NH and forms NH2 here, and there is rearrangement of hydrogen from this carbon to this carbon, and CH double bond CH is formed here. Thus this is cleaved as fumarate. So this fumarate is cleaved off from this structure and NH2 is formed here. Thus this AICAR is formed. Now in the next step, this AICAR in presence of AICAR formyl transferase forms FAICAR or 4-amino-imidazole carboxamide ribosyl 5-phosphate. Here transfer of formyl group from N10 formyl tetrahydrofolate occurs. So here this CHO group, that is formyl group, is attached with this NH2 group, and this hydrogen of this NH2, one of the hydrogen of this NH2 leaves with this tetrahydrofolate. Thus this bond is formed here. This FAICAR is formed. In the next step, that is the last step of IMP formation, here FAICAR in presence of IMP cyclohydrolase forms IMP, forms inosine monophosphate. So what happens here is there is removal of water molecule. That is, this one of the hydrogen from this NH2, this hydrogen from this NH, and this oxygen from this CHO leaves as water molecule. Then there is bond formation between this nitrogen and this carbon, and it is shown here. Now for maintaining the valency, a double bond is formed in between this nitrogen and this carbon. It is shown here. Thus IMP or inosine monophosphate is formed. So this is the pathway, first part of the pathway, that is the formation of IMP, and in the next video we will be discussing about the remaining part of this pathway for the formation of purine nucleotides.